Hello, this is Dr. Dan Guerra coming to you from VeraMed Studios in the Pacific Inland Northwest of the United States. Memorial Day weekend, in fact it is Memorial Day today, the 28th of May 2018. Today we're going to cover some ground of a paper that I picked up on one of my numerous news feeds, and it relates to the examination of a neuroimmune axis with a disease that's normally linked to autoimmunity, which means it's linked to inflammatory responses, and whether or not that inflammatory association of that disease, it's rheumatoid arthritis we're talking about here, human RA, rheumatoid arthritis, is linked to certain psychiatric issues, and the one that they're going to talk about is major depressive disorder. This is a study done in Korea. It doesn't really change the experimental design, any of the other methods, the data, or the description of the data, but it is a Korean population. And because of that, there'll be some parameters. You'll see some biometric parameters that'll be different than if this study was done, let's say, in Western Europe or in the United States. And I'll point them out when we get there. Let's go to the next slide. So, oh, by the way, that's me. I'm Dr. Dan Guerra. You can see me down here too. There's my email address. If you want to contact me directly about any of these videos that I present for VeraMed. VeraMed is my company. And what we do is we go to the basic scientific research, published research that has gone through our referees so that it has already been vetted by other scientists to be worthy of being published in our august journals, right? Our periodicals where we take our research from the lab bench and describe it to other scientists and to the world. So those are the only papers that I like to cover in VeraMed. Nothing popular press, no newspapers, no ad hoc reporting, none of that. This is science. And our company takes the science and delivers it to you after you propose questions or even premises that you would like to have evaluated, summarized, or interpreted relative to your own research interests or your biomedical interests. So primarily we deal with biomedicine because I'm a lipid biochemist by training. I have a PhD in biochemistry. But we also cover lots of other ground in biomedicine. So cell physiology, for example, genetics, epigenetics. We also, because of the co-founder, we also discuss pharmaceuticals, drug interactions, drug mode of action, and how that becomes part of a therapy in a biomedical program. So we also have a very strong issue with that. So without taking much more time talking about VeraMed, there are videos where you can learn more about our company. Let's go ahead and get started with this talk. So the question I pose here is, could neuroimmune signatures proxy endophenotypic correlates in psychiatric behavior? Okay, so you have to unpack a bit of that, but I think we already have an idea where I'm leading to. In other words, if we look at neuroimmune signatures, meaning like cytokines, for example, or chemokines or growth factors, these would be primarily proteins, and let's say we look at in the sera, can we say that those, the concentration and the appearance of those molecules proxy as an endophenotype for, let's say, major depressive disorder as associated in this particular paper with another disease, and the other disease is the autoimmune rheumatoid arthritis. That's what we're getting at here. By the way, there's my Facebook page. All of my VeraMed video lectures that I finally put into YouTube are all there. And you can contact me through the Facebook. You can contact us through the company site, which you'll see at the end, or via my email. All right. So low-grade inflammation has been suggested to decrease emotional recognition, that is, noticing emotional state or affect in others. The evidence in this is called the vaccination model of inflammation, and the paper that we're looking at is right there, Brain, Behavior, and Immunity, published just this month, May, in 2018. In fact, we're using the corrected proof, so this is yet to actually come up in print. Now, this diagram I show here is not from the paper, but it's basically what you need to know as prolegomena before we get into what the reasons for this research were. So what the people who wrote this work and did this work were thinking about is this model, that inflammation has been associated with a decreased neurotransmitter metabolism, decreased neurogenesis, and an increase in glutamate excitotoxicity, which means that rather than having inhibitory neurotransmission, you have an increase in excitatory neurotransmission. So all three of those are paradigmatically associated with certain inflammatory responses in mammals. Now, I will give a big caveat here. A lot of this research is done in animal models, mice, particular rats. But this has been shown, these three paradigmatic associations have been shown linked to inflammation also in human studies. Now, what all that can lead to is an altered neurocircuitry, obviously. So, for example, in the anterior cingulate cortex and in the basal ganglia, important areas in the brain that are associated with fine motor function, as well as the link between affect and cognition via the prefrontal cortex. Ultimately, those kinds of signatures can lead to, with this model, major depressive disorder. Okay, so from inflammation through an endophenotypic screen, culminating in one final reorganization, you could call it a pathological reorganization or altered neurocircuitry, leading to major depressive disorder. So, what does the paper want you to know as they introduce it? They want to say it's becoming an accepted hypothesis that inflammation may affect social behavior. So they're trying to link just generally, if you have an inflammatory response in the body, it may affect your affect, or that is your emotions. It affects your emotions because your emotions are linked from the limbic system to the prefrontal cortex and the occipital lobes to the way that the particular person who has an inflammation behaves. That is, for example, a classic case would be irritability. Let's say you have a sore throat, you have inflammation in that part of your body. Well, you're going to be a little bit, upset about that. It's going to hurt. It's not going to be so easy to swallow or to consume food. You may be running a low fever. There might be a lot of other things associated with that. Well, obviously, it's going to make a person irritable to be irritated by that inflammatory response. So that can directly affect your social behavior. Just imagine how you feel when you get up in the morning and you notice you have a sore throat. It's going to be a different day than if you woke up and you felt perfectly fine, or as fine as you can feel, depending on who you are. All right, so that's one just generalization, right? Second thing, inflammation correlates with feelings of social disconnection, and it may even sensitize behavior to social threats or rewards. So this is getting deeper into the psychiatry of the system. So they're trying to say that simply because inflammation is occurring, you may also have a problem interacting with others. You may not just feel bad, but you have a problem with others, like you may get more upset with them. Furthermore, having an inflammatory response in the body may even sensitize you as to how you respond to other people, such as a social threat or a reward. It may blunt a reward feeling, or it may enhance or excite a social threat feeling. Like you feel like somebody's talking about you, somebody's putting you down, somebody's staring at you. It may enhance your response to that in behavioral ways that might be negative for both you and the person that you're responding to. Okay. So research does suggest that inflammation might diminish the capacity to recognize the temperament of others. Here's another component. What are other people thinking? Well, if you have some kind of inflammatory response, you're feeling irritated, you might feel very sick, in fact. You might also think about how other people treat other people. That is the question of other minds. So you might have a problem recognizing what you think is some kind of social decoration of other people's minds, or that is recognize the temperament of others. And that may be linked to parts of the brain that notice facial expressions. We have a very important locus in the brain that notices people's faces and how they respond. Happy, sad, concerned, conflicted, contemplative, all kinds of ways that people can express themselves just with their face, right? Can help you as the interpreter of that other person that you're observing to determine what they're about, what they're thinking about. Well, it seems to be that this inflammation response, if you buy into this theory, can diminish your capacity to do that, to tell what other people are thinking about or how they're going to respond to you. And that comes back into that threat mode we talked about. So basically that's called a loss of activity for reduced capacity of theory of mind. Theory of mind is what are other people thinking? What are other minds thinking? It's called theory of mind. We all have one about the world about us. So the concepts of theory of mind is an empathy measure, and we call that mentalizing. And we get a lot of this research from studying autism spectrum disorders. So it's been observed that this theory of mind alteration seems to also be found in major depressive disorder. Now, ASD and MDD don't carry many neural correlates. There are some. But so the endophenotype in terms of biochemical interactions are there, and they seem to be woven together in certain portions, certain nuclei in the brain. Again, limbic and prefrontal cortex, the executive decision-making process and the emotional process. The limbic is the emotional or affect, and the prefrontal cortex and associated conduits back and forth in the ascending pathway are linked to emotion, which is primarily the limbic system, things like the amygdala. Okay, and also other portions of the brain that link to motor operation, such as basal ganglia and memory. Okay, so the data meta-analysis that these people have looked at seem to support a connection between major depressive disorder and inflammation. So these authors are speculating, which is usually not a good thing. So I want you to get an idea right away that I'm not really that happy with this paper. I'm not saying it's a horrible paper, but I chose to go ahead and show you this paper and pull it apart for you on this video so that you get an idea of how research can be misdirected. Okay. All right. So anyways, it might warrant careful scientific analysis. So let's continue on here. What's theory of mind? Well, you have affective theory of mind and cognitive. So affective has to do with understanding one and others' different perspectives and emotions, right? Remembering one's and others' different perspectives and emotions, and then using that perspective. So what kinds of things feed into that whole system of theory of mind of affect are emotional information guides, processing and regulation, cognitive flexibility, whether or not you can think clearly through something or you just immediately respond to an excitatory state, your working memory, what has happened before, and any kind of inhibitory control. So here's where executive functions come in, okay, feeding into the limbic system. There's also a cognitive theory of mind, understanding one's own and others' different perspectives, intentions, and beliefs, right? This is different than emotion, right? And how that information is processed and using others' perspectives, intentions, or beliefs to make inferences rather than one's own. So thinking about how other people are interacting, thinking about how you're responding to it, and thinking about how that collection of ideas may interact with one another. And again, the executive functions here are working memory, inhibitory control, and cognitive flexibility. And we get this from this paper here. There's the MedSci paper 2017 citation where this particular. So what is this paper going to talk about? It's a current cross-sectional study, and here are the aims. What is the prevalence of MDD in patients with rheumatoid arthritis? Remember, that's an inflammatory response, inflammatory disease. What are the risk factors for aggravation of depressive symptoms in patients with this other disease, RA? And what, if any, is the relationship between depression and disease activity? Proinflammatory cytokines like tumor necrosis factor alpha, interleukin-1 beta, interleukin-6, and then various trophic or growth factors like BDNF, GDNF, IGF1, and VEGF. And so they want to look at both growth factors and pro-inflammatory cytokines and determine whether or not these play a role between any link that you might get associated between depression and having an autoimmune disorder such as rheumatoid arthritis, which actually is linked to a lot of pain. So for you to understand why they're looking at these growth factors, I'm not going to recover the pro-inflammatory cytokines, just so that you understand that these are secreted proteins, often from immune cells, but can also be generated really in any cells, such as epithelia. And these, what pro-inflammatory cytokines do is they set up an inflammasome, an inflammatory court, if you will, in tissues that are usually linked to try to destroy foreign pathogens, but can also be evoked upon stress to the system. So that's what and you get an inflammatory response. The tissue goes into various states that alters its normal programming. It can become autophagic, it can become apoptotic, it can become senescent, or it can signal to kill other cells once it recruits the acquired immune system, such as T cells, natural killer cells, things like that, cytotoxic T cells, and of course the B cell and complement. So that's all I'm going to say about pro-inflammatory cytokines, the fact that it induces inflammation. These are secreted proteins that signal and traffic other inflammatory systems to come to bear on given tissues. Full stop. Now, what about growth factors? Well, let's talk about them because these might not be as, something you're familiar with on a day-to-day basis. First of all, brain-derived neurotrophic factor. It goes a member of the nerve growth factor family of proteins. So this is a protein that seems to be associated with how or what stimulates neurons to grow. So what can we say about BDNF? Generally speaking, alternative splicing of that transcript results in multiple transcripts, at least one of which encodes a pre-protein that has to be processed ultimately to a physiologically mature protein. Very standard in any kind of polypeptide, this kind of pre-protein, but in this particular case, it probably has to do with the trafficking of the BDNF from the cytoplasm to various other situations, locales in the cell. The binding then of BDNF to its cognate receptor promotes neuronal survival in the adult brain. This has been shown. Expression of the BDNF is reduced in diseases such as Alzheimer's, Parkinson's, and Huntington's chorea. So that may mean that BDNF is necessary again for neurogenesis or neural development, because in all of those diseases you have a problem with that because you have neurodegeneration. Right? BDNF may also play a role in the regulation of the stress response and in psychology of mood disorders such as major depressive disorder. What about GDNF? That's a glia associated. Okay, so glia, remember, those are the non-neurons, but still important cells in the central nervous system. So it's a glial-derived neurotrophic factor and encodes secreted ligand of the TGF beta, that's the transforming growth factor beta superfamily of proteins. So it's part of that entire family of proteins. GDNF binds various TGF beta receptors, leading to a recruitment and activation of another class of proteins called SMADs. And those are basically transcription factors, so as they induce the expression of genes at a specific locus, and that then therefore GDNF regulates gene expression directly. GDNF pre-protein, again, proteolysically processed to generate each subunit, and then you make a homodimer with disulfide linkage. I don't know if that's important, but that's just more detail about what this protein is about in terms of its chemistry. GDNF may also promote the survival and differentiation of dopaminergic neurons in culture, and it may be associated with motor neuron degeneration induced by anoctomy. So that's when you cut out axons, you need to have a regrowth or regeneration of that neural thread, and it seems like GDNF is necessary for that, in particular in loci that are rich in dopaminergic neurons. GDNF mutations may be associated with Tourette syndrome too, so this has been suggested. So those of you that know anybody with Tourette syndrome, they may have a problem with GDNF. Again, it's an association, it's not a causation. All right. Now, what about IGF1? I stands for insulin, so it's an insulin-like growth factor. It's structurally and functionally related to insulin, but has a much higher growth promoting activity than insulin. Remember, insulin is the major hormone from the pancreas. IGF1 it does things similar depending on its reception in peripheral tissues. But IGF does a lot more, and so that's why we call it a growth factor. It stimulates glucose transport in bone-derived osteoblasts, for example. It's effective at much lower concentrations than insulin regarding glycogen and DNA synthesis and glucose uptake. Those are all things that insulin, by the way, covers. It could play a role in synapse maturation, very important in neurotransmission then. It's also calcium-dependent exocytosis of IGF1, that is when you release IGF1 secreted, and that's required for sensory perception of smell in the olfactory bulb. You see, we're starting to get a really broad understanding of this growth factor. It binds to an alpha subunit called the IGF1R, leads to the activation of the intrinsic tyrosine kinase activity, so it autophosphorylates itself. And once that's done, it instantiates or initiates a cascade of downstream signaling events leading to the activation of the phosphatidylmethyltransferase 3 kinase, AKT-PKB pathway, and also the RAS-MAP kinase pathways. If you don't know what those are, please go back and watch other previous talks I've given in VeriFmed, or you can just look them up or Google those and you'll be able to get an idea. Basically, those are all kind of collectively, you could say they're involved in protein synthesis. And some of these particular cellular pathways include angiogenesis and anabolic growth, right? So IGF1 also binds to integrins directly, and that promotes extracellular matrix adhesion. So there's something to do with cellular trafficking. Okay? So just to get an idea of how these growth factors can work, there's AKT. It blocks this tuberous sclerosis protein, which would normally shut down this GTP system, this RAS GTP system. So when you block this, you don't block this pathway, so you continue on. When you do that, this complex with SEC5 and XOB4 turn on mTORC, that's target of rapamycin, the cellular target of rapamycin complex 1. That's going to block autophagy, and it's going to turn on protein synthesis via the phosphorylation of this transcription factor S6K. Okay? It's also going to be involved in amino acid metabolism, which is going to be necessary for protein translation. Right? So this is just one of the embedded pathways that are turned on by IGF1. That's what we're showing here. All right. Now, an important note on IGF1 in clinical major depressive disorder that you'll find in the literature. This comes from a review article, Annals of General Psychiatry, published last year. IGF1 levels in MDD patients, major depressive disorder patients, mismatch across studies, and that may arise from various factors: age, gender, course of the disease, that being MDD, presence of cognitive impairment, and ongoing therapy or general health conditions. Okay? Initial peripheral IGF1 levels may predict the occurrence of depression in the future. This is an argument as using it as a biomarker. Peripheral IGF1 levels may reflect cognitive dysfunction as well, although the data is quite limited. It's there. It's also difficult to evaluate, though, the influence of treatment of IGF1, since there is variation at baseline, although most of these papers showed a decrease in IGF levels after treatment. Okay, so what I'm saying here is that you go through this whole review article and you see that there's a lot of ambivalence as to whether or not IGF1, just the concentration of its availability, its occurrence, showing up in a cell, really has a direct, always one linear effect. Okay, doesn't seem to. But that's common with growth factors. Varying concentration changes the quality of the response. So quantity is finally read out as quality. All right, finally, VEGF-A as the vascular endothelial growth factor. It induces proliferation and migration of vascular endothelial cells. It's essential for both physiological and pathological angiogenesis, so really important as a vascular system. That's why it's called VEGF. Disruption of that gene in the mouse model resulted in abnormal embryonic blood vessel formation, as you might guess. VEGF-A, the isoform we're talking about here, is upregulated in tumors. Its expression is correlated with tumor stage and progression. Elevated levels are found in patients with POEMS syndrome, that's polyneuropathy, organomegaly, endocrinopathy, monoclonal gammopathy, and skin changes. That's why it's called a syndrome. It has all of those component pathophysiologies. The allelic variants of VEGF-A have been associated with microvascular complications of diabetes 1 and even atherosclerosis. Alternatively, splice transcripts of variants encoding different isoforms have been described. Okay. It's active, as we said, in angiogenesis, vasculogenesis, and endothelial cell growth, as the name implies, infers. It induces endothelial cell proliferation, promotes cell migration, inhibits apoptosis, and induces permeabilization of blood vessels. You can see how VEGF might be a problem in cancer. Blood VEGF levels in patients with MDD are significantly higher than healthy controls. That's way down at the confidence level of 0.001. And so it may be a disease marker in patients with MDD. And I got that particular point from European neuropsychopharmacology paper some 3 years ago. Not quite 3 years ago. All right. Let's continue on with the paper then. I gave you the background on growth factors. How do they assess RA? Now this is kind of a very difficult, overly complicated, I think, set of parameters that these authors like to use. But I have to bring it out because you're going to be looking at data. You're going to say, what does this mean? So now I'm telling you this, and you can refer back to it. Disease activity score 28 joint counts with a protein called C-reactive protein, which is usually linked to pain, nociception system. So that they call that DAS28-CRP. I don't like that because, yeah, okay, it gives you the first letter, and so it's an acronym, but it's really, I think, a pretty lousy acronym. So that's, again, an assessment of RA. And the routine assessment of patient index data 3, which is known as RAPID-3, both of those were used to evaluate disease activity. So both of those, when you have high values for that, means you have high levels of this autoimmune disease, rheumatoid arthritis, basically. DAS28 includes swollen joint counts, SJCs, tender joint counts, TJCs, of those 28 joints that they measure. The CRP itself, of course, and the patient's global assessment of disease severity, that's usually with a questionnaire, and they call that GH, on a visual analog scale of 0 to 100 millimeter. Okay, so these are complicated readouts, but I want you to get an idea of at least how they come up with the numbers. So the DAS28-CRP is scored 0 to 9.4, with scores less than 2.6, indicating disease is in remission. Between 2.6 and 3.2 for this value, you have low disease activity. You can see the intermediate, and then you see with high disease, it's anything over 5.1. Okay. The RAPID-3 includes a subset of core variables via assessment questionnaire, which I alluded to above, and scores 0 to 10. Okay. Scores 0 to 1 indicate disease remission, 1.1 to 2 low, 2.1 to 4 moderate, anything above 4 all the way up to 10. It seems like a broad range out there at that far extreme. That's high disease activity, and this is all about rheumatoid arthritis. So you can see the values here. Here it is for the people with depression. That's the number of patients. People with a not depression, this is their depression scale here. That K stands for Korean, and the rest of the scale they get directly from associated parameters from DSM-4 and 5. And you can see that there's a difference here. Okay, so there's 26.9 plus or minus 9 versus 8.1 for the disease. Okay, for depression. Well, that's how they're describing it. These people are depressed. These people are not. Okay. Everything else, okay, depression versus non-depression. You can see how it plays out. Here's gender, okay, but even though you have 121 females here versus 254, there's more people. So the percent of females, the age distribution is in the low 60s. Education is similar. That looks like it's not even quite high school, which is interesting. The BMI is real low. There's one of the parameters you would not get if you were doing this study in the United States. It's a really nice low BMI. These people are healthy weight. In fact, they're probably thin. Smokers, there's not very much difference between the two. They seem like there's more smokers that are depressed than not. Heavy alcoholics. It's funny the way they came up with that, like how many drinks per week, and they call the person alcoholic. I don't know if DSM would agree with that, but whatever. Okay, it seems like there's less drinking in the depressed people and more in the non-depressed. Now that's also kind of interesting. Maybe those people self-medicate, right? Okay, so some of the other things, living alone. There's more people who are depressed live alone. Income, more people who are depressed, they have a lower income. Hypertension, there's a little bit more hypertension in depressed people. Diabetes, it's about the same, so that's not going to be a conflicting morbidity interactor. And fatigue, they're both saying they're about similarly fatigued. So MDD was estimated using the Korean version of this Beck Depression Inventory, second edition, so they call it the KBDI-2, and that's commonly used in Korea to diagnose depression, and also it gives you a severity scale. Questionnaire contains 21 questions regarding emotion, cognition, motivation, physiological and other symptoms. The score greater than 18 is considered cutoff value for depression according to the previous validation. So 26.9 is well above 18, so these folks are, I guess, depressed. All right. Now, here we're looking at all of various parameters here. Here are autoantibodies, the IgM-RF, and the ACPA, which is the anticyclic citrullinated protein antibody. CRP, of course, is C-reactive protein. These are proteins that come up when you get an autoimmune response. They increase in autoimmunity. Okay, and so what you see here, there's really nothing different between the depressed and non-depressed. Okay, so the disease severity of rheumatoid arthritis is not there. So there's no differences in the rheumatoid arthritis associated autoantibodies between depressed and non-depressed. And all these other things are just telling you whether or not they take drugs. So DMARDs are disease modifying anti-rheumatoid drugs. GF is again the patient's global assessment, and none of these really differ between the depressed and non-depressed. So just telling you these are the set points, right? And this is what you want. You don't want to have big differences here because that could then conflict with and cause problems with what data you might be able to get as a readout. So patients with depression had, in general, lower education level and income. They tended to live alone, as I pointed out, and they had higher disease activity as assessed both by that RAPID-3, but only slightly higher. Okay. Another important point is a significant correlation was found between those two disease correlates in depressed patients. So there's an interaction between those two ways of analyzing rheumatoid arthritis. They used a multiple regression analysis, and they looked at the severity of fatigue, the RAPID-3 score, there's their odds ratios, and all of this they took together independently related to depression. It's the way they ran their correlation. Okay, so what do you see here? Look at the difference between these high disease, okay? Here's the high rheumatoid arthritis, and you see here it's about what? Twice as many people who are depressed, right? Okay, so what does all this tell you? A lot more people down here in the high range. So it tells you that people who have rheumatoid arthritis and they have severe disease are more depressed. Well, wow, that's really surprising, right? Of course it isn't. If you have a really bad case of autoimmune disease, you might be more depressed. That makes a whole lot of sense. So 59% versus 27%. Don't worry about these raw numbers. They look similar. That's because the N number is different. Okay. No surprises. That's really all they can say. So I'm not going to show you the data because it's rather boring, but there were no differences in the pro-inflammatory cytokines between depressed and non-depressed patients. There were subtle, but it was a statistical decrease in BDNF in the depressive patients. Okay. So, but again, I'm not very much impressed with that. These are serum markers too. So, and then in there, I think lies the problem. So what they summarize, let's summarize for them. RA patients in Korea were more depressed than general population. Gee, okay, 32% versus 6 to 8. So I guess if you talk about how many people are depressed, and this is of course South Korea, not North Korea, I want to point that out. Only about 6 to 8% of South Koreans claim to be depressed. Whereas if you have rheumatoid arthritis and you're Korean living in South Korea, it goes up to about a third, right? Okay. There's more joint pain, physical distress made people more depressed. Duh, right? One would guess that might be the case. Depressed people had no differences compared to non-depressed in serum pro-inflammatory cytokines. So that's a bust for this paper in a way, right? Depressed people had no differences compared to non-depressed in all those growth factors I went through, except maybe they have a lower BDNF. Okay, so maybe there's some issue with neural repair and neurogenesis, but lower doesn't necessarily mean the activity is lower in the tissues that would need to be represented for BDNF to actually have a causal or even a highly correlated link between depressed and non-depressed RA people. So let's ask the question, was this experiment even successful? I'd say it's hard to assess given the lack of examination of immune cellular activity. So we need to know what was going on with things like macrophages, M1, M2 classification. We want to know what's going on with dendritic cells, the trafficking of antigen presentation to the acquired immune response. Of course, we'd want to know about natural killer cells, a topic in a previous talk from Vera Med. We'd like to know about cytotoxic T cell activity, about other T lymphocytes, the Th1, Th2, Th17 responses that I talked about, B cell metabolism, mast cell metabolism. Okay, these are all things that were not covered. There was no cellular data at all. Hard to assess any of this without taking a look at the cells themselves, populations, and what they're secreting and interacting with. Same for growth factors relative to subsets of immune cell proliferation. Where were the growth factors? This is serum growth factors, where they linked to T cell populations, B cell populations, the innate immune response cells. None of this was tried. RA is an autoimmune disease. So any extra associated biochemical readout from that could be simply masked or underdetermined, I call it, because depression itself, right, may be linked to that inflammatory disease. And so how do you really separate the two? Okay. Plus, you're talking about what? You're talking about inflammatory markers. So you already know you have an inflammatory disease. So how are you going to look at something above a superannuary to the amount of inflammatory cytokines from baseline, or baseline being you are rheumatoid arthritic? Okay, that's an issue. So what else I want to say? Major depressive disorder associated upregulation of anti-inflammatory corticosteroids, which we know happens, could actually counterbalance cytokine levels. So you get this immune suppression because you get a buildup of cortisol during depression. Been well documented. Negative results are just as important to assess overall pathobiology of the system. So I put that in there, I put all this in here, because I'm saying, well, even though we didn't see anything, that's important to put in the literature, right? It's good that we know now that not every time you look for the induction of pro-inflammatory cytokines in depressed people that you see a jacking up of the concentration of those in the serum. It's not the case here in this study. Again, different subset population compared to the West. These are South Koreans. May have something to do with how they interact with pain, how disease severity is marked and cataloged, and indeed how major depressive order is looked at, even given cultural differences. All of that has to be put in as caveat, even though it may not be a caveat. At least think about it. What does the published literature say about this? Okay, so I went and looked, of course. I pulled down about 140 papers just by a quick search in PubMed, and those papers were published just in the last 10 years. And they seem to link depression to serum cytokines, what this paper was hoping to do, but the majority showed variable results. When I went through those papers, and I went through dozens of them, not every single one, you have high versus low versus no correlation of pro-inflammatory cytokines to depression. So it kind of runs the gambit, right? Neutral, positive, negative. So that's not very reassuring that pro-inflammatory cytokines are going to be always linked to major depressive disorder, because I just don't see this in these papers. These are all current papers vetted and published in the scientific literature. Here's 3 Journal of Affective Disorders published in 2018, just last month. They found no correlation. Molecular Psychiatry, published about a year and a half ago, was a review article. They suggested some uptrending in pro-inflammatory cytokines in MDD and in bipolar and even in schizophrenia that was reduced upon treatment. But the overall picture was not very convincing. I mean, I read the whole paper, and again, I came up with a lot of ambivalence. The numbers were not that striking, although general trend, some uptrending. Okay. Finally, psychiatric research published again just very recently, about a month and a half ago, has shown that pro-inflammatory links to adolescent abuse and subsequent MDD were there. Now this is looking at now children who were abused as adolescents and how they turned out as adults or young adults. It seems to be a link between pro-inflammatory serum markers and major depressive disorder. So here, no correlation, and a positive and a negative correlation. All three of those seem to be on the plate. So let's do the dialectic, right, across the discussion. I argue that rather than a general theory for inflammation and any neuropsychiatric state, including MDD, bipolar, etc., the researchers need to examine the genome, the environment, and the epigenome of all the individuals in the research study with the disease in question. And with that look-see, you may be able to get more precise, okay? So the look-see means what's going on with the genotyping of the people that you're looking at with major depressive disorder and with any associated inflammatory response or growth factor response. What kind of environment are these people in terms of not just environment like where they live, how old they are, age distribution, gender distribution, but also how they're aging, health otherwise are all common, very common comorbid responses in terms of how much inflammation could be linked to neuropsychiatric conditions. And of course, all just, how that environment endogenously, the endophenotypic environment, that is, are there a lot of autoinflammatory diseases, for example, in these people? Do they have chronic illnesses? Do they have episodic acute illnesses, right? Do they get a lot of common colds? Do they get a lot of sore throats, like at the beginning of the talk when I talked about? So that's all about this new theory I'm putting out, called diaeventomic, which tries to link, we're working to link the genotype, the environment, and the epigenome to discussions of the neuroimmune axis relative to overall human biomedical condition or state. Okay, that's very briefly what diaeventomic is about. So we talk about it being dia, meaning across or beyond or furthering an interactive discussion, and I call it an eventomic response because these are not substance responses. So when we look at individual concentrations, for example, of pro-inflammatory cytokines, it doesn't tell us anything about its half-life. It doesn't tell us how long the pro-inflammatory cytokines have been coursing through the veins of the people that have high, low, or intermediate levels of those particular substances or growth factors, and even when you look at the cells. So you need to look at the dynamics of the system, and so it's an event ontology, not a substance ontology. Event meaning we have to look at how things change over time and over space in order for you to get a real handle on how genomics, environment, and epigenomics interact in that triplanal grid, right? And if we don't have that kind of information, it's going to be very difficult to draw serious conclusions. So I say that diaeventomic studies assessing individual, one patient at a time, immune cell populations, single nucleotide polymorphisms just in the genome, the methylomics and the cytolomics, that is the epigenetic markers that we normally look at, serum peptides such as pro-inflammatory cytokines, growth factors, other proteins, for example, involved in metabolism or endocrine system, and overall, as I said, comorbid diseases, with all of that linking with mood disorders that have been clinically diagnosed, of course, and then the clinical therapy that may have been driven up against those mood disorders or against any of the other diseases that may have been employed, such as even things like immunizations and taking of things like non-steroidal anti-inflammatory drugs. All of that can be done on an individual patient basis. Until we start doing that, I think making broad sweeping generalizations is always going to run up against a wall of basically prevarication. How much of this can we say is actually true, right? We don't know because there's a lot of ambivalence, there's a lot of variation. So in order to get away from that variation, get away from believing opinions and actually understanding at the level of conceptual knowledge, we need to get more data. And the data not only has to be correctly produced, that is, design good experiments, but also it has to be vetted well. And then from that, go back and take that data, the vetted data, and take it back to the scientific literature and use that as foundation and coherence to be able to come up with some truths that we can justify, that ultimately can be believed and turned into knowledge. Now, I'm waiting for this paper to be lifted on embargo. I was surprised just because this paper doesn't come out till June. I mean, here it is, the end of May. I mean, I'm not all being, pushy about this, right? I'm not being impatient at all. But I got to read the abstract of this paper and a little bit of the first couple of paragraphs. That's all they give you as a little snippet of the paper. But I have to wait for the embargo, even though we have full access to this journal. It's Nature Immunology. Go figure, nature's like that. Paper will come out in June, and there's a citation. Now, I like this paper because this one's going to be, I think, getting more down into diaeventomic parameters. So we're going to probably review this paper, or at least put in a collection of reviews, some subsequent VeriMed video. This I can promise. So what does this paper say? Why am I intrigued about it? It's talking about the polarization of macrophages into M1, M2. M1 macrophages are pro-inflammatory, so the way that they work as antigen-presenting cells and as authentic card-carrying macrophages is different from M2, which are basically anti-inflammatory macrophages. So those tend to be working in contradiction to one another. And how you turn M1 into M2 has to do with what's going on in metabolic pathways inside those macrophages. Yeah. Which means how signaling works, how biofuels are interacted, whether or not you're using fatty acid and glucose, for example, amino acid even. It's unclear, this paper is going to show eventually, I hope, how or if mTOR, remember it's the target of rapamycin, regulates metabolic status to promote polarization of those macrophages. Remember, mTOR is involved in a global protein synthesis, anabolic system, right? Unclear whether or not that's going to play a major role, which means we're going to look at that embedded pathway and it's going to make sense here. However, the mTOR semaphorin, which is going to be a target of discussion in this paper, called SEMA6D, proximal proliferative receptor gamma, PPAR gamma axis. So you've got mTOR, SEMA6D, and PPAR gamma, that three-way axis. I always like three-way axis because you get a lot more fundamental interaction that way. Could play a vital role in macrophage polarization. So if you look at all three, you start to get actual information. Remember I told you how quantity, when you start racking up quantity and changing concentrations of things or changing the number of interactants, can lead to a quality change. Very important to consider this concept. So, for example, inhibition of mTOR or loss of SEMA6D blocked the anti-inflammatory macrophage polarization. So all you had was the inflammatory. That's not going to be good in what? Autoimmune diseases. And that's concomitant with severe impairments, ultimately, to the PPAR gamma expression. That's going to be a transcription factor involved in lipid metabolism. And there you go. Uptake of fatty acids and lipid metabolic reprogramming is going to be altered because of inhibition of mTOR or loss of SEMA6D. So you see how this quickly becomes more complicated once you start laying down further foundations that can cohere relative diaeventomically to the disease state you're trying to look at. Here we're just looking at switching M1, M2 in autoimmune diseases or in just normal inflammatory responses. One cell type here. In fact, macrophage expression of the receptor plexin A4 is responsible for the SEMA6D mediated anti-inflammatory polarization. We've got other things to worry about. Here's a receptor protein, plexin A4. We need to also bring it to the mix. Indeed, SEMA6D is important for the generation of intestinal resident chemokine receptor high macrophages and prevents development of colitis. So here we've got an inflammatory disease of the colon, colitis of the intestine, and that seems to be linked with, again, the SEMA6D in macrophages using a chemokine signaling pathway. So not just cytokines, but chemokines, right? Sure. And all of these growth factors and indeed the stable core intermediary metabolic shunts such as PPAR gamma and mTOR. So this suggests a seminal role. That's what this paper is going to talk about. That's why I want to really get into it. I like the fact that it's SEMA 6D, see, seminal role. My little haha humor for today. Reverse signaling, that means turning things off or repealing them. That would be lipid metabolism in this case, which of course that's why I'm interested in it, because lipid metabolism is the most important metabolic pathway, full stop, in every living system and also in dying systems. Okay. In macrophage polarization, immunity and metabolism via the PPAR gamma. Okay, so this is going to be a tremendously great paper to dissect. I'm planning on doing it very soon. Just want to bring that up as here's a better version of the way of getting at the answers of the things like, is major depressive disorder linked to pro-inflammatory cytokines in an autoimmune patient cohort? So this is the kind of paper, see, it's not talking about any of those things that the last paper did, but this is the kind of paper you can use to be able to derive more information and get better experiments maybe for the paper that for the kinds of papers we just looked at. All right, so there I am at the end. There's our email address for VeraMed, info@veravmed.com. Please contact us there if you'd like to be our client, and also the my particular email address is there, or you can always contact me about the content and further content and composition of these talks. I'm willing to take interested parties on what you'd like me to talk about. There's our website. Please go visit it. We're a very stimulating young company. And again, we are scientists verifying published evidence in medical biosciences. So I'm going to do my sign-off today, a special sign-off today because it is Memorial Day. I want to thank all the people who have risked their lives and those poor unfortunates who have lost their lives whenever there has been conflict anywhere in the world. Particularly, I want to think about those of us in America who have lost loved ones either in war or suffered because of consequences of war, and that's a lot of us, I know. So hats off to them. Thank you very much for paying attention on this rather subtle Memorial Day, and may you have an enriched and meaningful and authentic Memorial Day weekend, what's left of it. But all I'm going to do now is sign off and say bye for now.